Video. So let me see if I can do live talk. If I could share my screen. Just one second.
While Gianfa is setting up, I will quickly introduce myself. I'm Gina Baucom, and I'm the moderator. I will be telling each speaker at 12 minutes past the start that it's 12 minutes past and you have 3 more minutes.
All right, great. Thank you. So again, my name is Xianfa Xi. I'm associate professor at the Department of Biology and also director for the Center for Biotechnology, Genomics, and Bioinformatics at Virginia State University. So good evening, good morning. Actually, right now I'm in the complete different time zone, 12 hours ahead of the Eastern Standard Time. So forgive me for anything that make me look a little bit different. But anyhow, so it's a really great pleasure to talk with you today about a topic that's been interested me for a long time. That's the role or relationship between epigenetics and evolution. Let me move on to the next slide. So again, this interest has originated when I was a graduate student, but of course at that time this might be considered as a taboo because if you think about epigenetics, you were probably considered as a Lamarckian. But in 2004, I give a presentation with the title "The New Evolution." Since I know the title looks too big, but what I tried to do over there was try to integrate epigenetics, dynamic evolution, and Evo into evolution biology. So in 2009, I organized and chaired a symposium on this topic with the title "Epigenetics, the Environment, and Evolution of Triple E's." So even though this happened at the SMB meeting, but I believe this was probably the first one, the first symposium dedicated to this topic, epigenetics and evolution. Of that, I know there had been some other symposia in either evolution meeting or SMB meeting, and there have been a lot of publication on that as well. I myself also was involved in some research looking at epigenetic differences, genomic differentiation, as well as transcriptomic differences between cultivated rice and wild rice. If you're interested, you can look at this paper. So before I try to answer this question, whether epigenetics really plays any role in evolution, first I want to be clear what I meant by epigenetics, because this term has been used in different ways in the past and today. So it was Conrad Wellington first used this term epigenetic, but he used this term to describe the situation that from the same genotype, you could have different phenotypes. So this is different from how the term is used today. So this is on the right in blue. This is the definition I would like to give for epigenetics today. So in my opinion, it means trait change and inheritance not due to change in the primary DNA sequence. So some people have made this term pretty broad, including both behavioral inheritance and cultural inheritance, but I would like to focus on cellular epigenetic inheritance. So speaking of the mechanism at the cellular level for epigenetic regulation, so usually happen into formed DNA methylation, which usually happen to the cytosine, and histone modification, which happen to when different residues in the H3, H4 histone molecules in the form of methylation, acetylation, phosphorylation, or in some cases binding to another protein called the ubiquitin. So the combinations or the different combinations of this modification that would like the and And epigenetics today, probably we can look at this theory. So given that the alteration of epigenetic states of genes could alter gene expression, which in turn could alter developmental or metabolic pathway and produce new phenotype. And also given that epigenetic state changes of transport element and tandem repeat could alter their activity, which otherwise could cause large-scale dynamic rearrangement events or drive dynamic evolution. And also epigenetic state could be altered by the environment, and some of those changes could be inheritable. Put all this together, so we believe that some of us believe that epigenetic inheritance may provide a mechanism for the inheritance of acquired characteristics. So if this is true, then the environment is not simply a filter like what the natural selection theory would propose, but it could be a real driver directly shaping the new phenotype or creating new phenotype in the offspring. So together with this, we see there are a variety of reasons to make natural selection power, I guess.
So
I got
just so you know, we could hear and see almost none of your talk, so we can offer you a slot at one of the options tomorrow. I will be in touch by email.
Oh, you couldn't hear me at all.
Basically, no.
Oh, I'm sorry.
It's okay. But it's now
Okay.
time for us to move on to our next speaker, actually.
Okay, all right then. Thank you.
Hello. So I'll just share my screen. Okay, so I guess I can start. Okay, so I'm Alan, and I'm glad to be here to talk about my work on the selective advantages of related helping in the viscous population. So one natural question that arises is, nice. So what is related helping? So related helping is an altruistic behavior that happens in a lot of different species of birds, so for example, the long-tailed tits, the western bluebird, or the Iberian magpie. And so if you consider an adult bird, it will lay some eggs, but sometimes a predator will come and eat all the eggs. So in the end, these adults will not have any offspring this year. So in that case, it can decide to help a fellow related neighbor to have either a higher survival or a higher fertility. So for example, by feeding the offspring, and which will have like a higher feeding success. So studying the evolution of related helping, while it looks like quite straightforward, like if the benefits of the help is like overcoming the cost of the help, then okay, it may just be good to help in that case. Thing is, if we look at a viscous population, where individuals are dispersing not far from the natal sites, individuals will help so relatives, but will also compete with them. And as you can imagine, it may not super good to help a competitor. And so in this presentation, I'll talk about how the viscous population will shape the evolution of related helping. And also I will emphasize the role of the spatial structure. So you'll see I will use a specific, an explicit spatial structure here. And also what are the impacts of the nature of the benefits. So for example, if we have some survival benefits or fecundity benefits, what does it change for the evolution of related helping? So to study this related helping, we are looking at one-dimensional stepping stone model, where we have individuals here will suffer brood failure, who will not have offspring, and some will have a brood success, so including here. And so individuals in red who would suffer brood failure will eventually help their neighbors by, so for example, increasing the survival or increasing their fecundity. And so after the offspring will mature, they will go out or stay there to compete for a breeding spot. So for example, here the individual will stay on the natal side to compete for the breeding spot with property 1 minus D, where D is the dispersal rate, and proportion D will go out in the 2 neighboring sides. So half of them on the right side and half of them on the left side. And then the offspring will compete for the breeding spot. So depending on the survival of the breeder, of the adults on the breeding site. So whether if it survives, then it will keep its breeding spot, but if it dies, the breeding spot will become available for the offspring to compete on. And so to determine which offspring has a breeding spot, it depends on the number of offspring competing for the breeding spot, and we just take one randomly from all this offspring. Now, how do we know if, how can we know if helping is good in this situation? So to do that, we compute the fitness, and here in the neighbor motivated fitness to see if it's so good to have helping. So first, with this fitness, it will depend on the neighbors in the neighborhood of a focal individuals that we measure the fitness. So for example, here, if we have our focal individual, its fitness will depend on the breeders who will eventually compete for this breeding site and the different neighboring breeding sites too. So we have to measure the fitness in every possible of our neighborhood, which depends also on which will take like 2 to 3 steps away from the focal individual. And so we compute the fitness of the focal individual given the specific environment. Then we sum all the possible fitness depending on the environment, and we multiply it by the properties that this environment is happening. So this gives us our total fitness of the focal individual. And now we can compute the neighborhood fitness change, so to see if it's positive or if it's good or not to help. And so this fitness change is basically the sum of all the impacts of the help. So basically on the different neighbors of the focal individual, including the focal individual itself, times the readiness between the focal individual and the impacted individual. And again, summing on all the possible neighborhoods of the focal individual. So what does this fitness change means? That if we have a selfish population and we measure the fitness change in the population, we have that if this fitness change is positive, then we have an emergence of the related helping, sorry. But if it's negative, then this related helping will not emerge. And so we can look at different scenarios. So for the first scenario will be when we don't have any brute failures except for the focal individual. And we can look at the different impact of the survival and fecundity benefits. And in the first plot here, we have the critical cost-benefit ratio associated with survival benefits. So just to summarize quickly, the critical benefits ratio, if it's high, then it will be easy to have emergence of related help. But if it's low, it will be harder to have related help. So basically, we need more benefits and less cost. And as you can see on this first plot, so associated with survival benefits, we see that for low values of survival, here on the left side, we have a low critical cost-benefit ratio. So that means for low survival values, it will be harder to have emergence of help, of related help, given survival benefits. But as we increase the survival, we see that it also increases the critical cost-benefit ratio associated with survival benefits. So survival benefits will drive the emergence of help better for high survival. And a surprising result we have is that as we increase the dispersal rates, so from the black curve here with D 0.2 to the blue curve D 0.8, we are also increasing the critical cost-benefit ratio associated with survival benefits. So this is quite surprising because if we increase the dispersal rate, we can expect that we decrease the readiness coefficient, which is true in our case. And so we can expect to have some so if we increase the dispersal rate, we decrease readiness, and so we expect to have a decrease in the helping rate, or at least to have more difficulty to have helping. But in our case, it's not the case because we also have this kin competition problem where basically if we increase the dispersal rate, we have less kin competition, so it's good for help to evolve in that case. So now if we have some fecundity benefits, we see a different similar pattern for the impact of dispersal rate, but we have a different pattern for the survival. And as you can see, when we increase the survival, we are decreasing the critical cost-benefit ratio associated with fecundity benefits. So for high values of survival, fecundity benefits will not drive well the emergence of help. And so here in that case, we can also compare survival and fecundity benefits by basically dividing these two curves together. And we see that as expected, for low values of survival, fecundity benefits will be better, but for high values of survival benefits will drive better the emergence of help. Now that we saw for low value of brute failure, what happens if we increase the brute failure probability? So in that case, we see we have that for brute failures that will be too large, too high. So basically, a lot of individuals will fail the brute. Then we can have related help may not emerge in that case. So all in this zero area here. But for low values of brute failure probabilities, then we can have fecundity or survival benefits driving the emergence of related helping. And so again, we have this difference between low survival and high survival. So for low survival, we have fecundity, which will drive emergence of help. But for high values of survival, it will be survival benefits. So except for this small area here. And if we increase the dispersal rate, this small area also increases for survival benefits. So what do the results means in terms of in a broader sense? So if we look at the spatial structure, so here we had a stepping stone model, but we also did a similar research on an infinite island model, and we found complete different relation with the dispersal rate. And in fact, so we have found the opposite relation. So this means that when we want to look at different behaviors, so for example here, an artistic behavior such as redirected helping, we need to be careful about the spatial structure we use, and especially for these dispersing patterns. And so this also is the case for different types of behaviors too. Another things we can say, given that we have different since the survival and fecundity benefits, is that depending on the life histories of a population, we may not have emergence of related help if some species may not be able to have a type of benefits too. Sorry. And so this life histories will will impact how different environments also will impact the emergence of this regulated habit. And finally, we know that helping can eventually have difficulty to emerge in the viscous population, but in our case, we also have some features that may make it more easy to have this emergence of help. So, for example, some overlapping generation, some in a way, some budding dispersal, because individuals are dispersing to different groups of individuals are dispersing to two budding spots together. And we also have some specific life history constraints. So with these root failures that individuals cannot have the root for a year, basically. And what can we do now, given our results and our structures? So one way would be to have a different specialized structures. So for example, special structures that would take into account that some bird species are making flocks of different birds before dispersal, which may again have some different consequences. And we can also imagine that given that this facultative helping behavior, we may have complete helping behaviors in future. So how this rotated helping will become complete helping in a way. So I just want to thank Jeff White, which is I think is listening now, and all the flower people from our Maths Bio group at Western for their useful advices. And thank you for your attention. And if you have questions, I'm still here, I guess, for 2 minutes, I think. Guess I can maybe stop sharing. There we go. Thank you for your attention.
Can you all see my screen? Is it working? Can you hear me? Just okay. So I think I can start now. So hi everyone, my name is Pedro Perez and I'm a postdoc at Florida International University in the United States. And today I want to share with you some of my preliminary results on the temporal genomic diversity changes of 5 pelagic deep sea species in the Gulf of Mexico. So usually when we think of genomic diversity and genomic structure, we tend to think of them in terms of spatial variation. So for instance, if we have site 1 and site 2, we can say that they share many alleles and they are connected through gene flow. Or maybe they do not share, they are sharing alleles, or maybe they are not sharing many alleles, so they are not that connected. Or we can even say that site 1 shows a higher genomic diversity than site 2. However, we can also think of genomic diversity in terms of time changes. So for instance, here, imagine that this is a population and the colors represent the genomic diversity. So we can imagine that there's a populational decline at some moment that will lead to a bottleneck event and loss of genomic diversity. And then the outcomes of this event might be maybe a recovery or maybe an extinction of that population. And of course, the outcomes depend on many factors. And when we think of genomic diversity changes over time, we can focus on changes that happened thousands of generations ago. So we can focus at changes during the last ice age, during the Pleistocene, or we can focus our attention to changes a few generations ago. And of course, in this second option, there are implications for conservation and management. And one of the groups that we least know about genomic diversity changes over time are deep sea species. And when I'm saying deep sea, I'm referring to that part of the marine environment that lie below 200 meters, encompassing all the environments beyond continental shelf depth, including both the ocean floor, but also the water column. And it's kind of surprising to think that we do not know much about population dynamics of deep sea species, because the deep sea is the largest environment on Earth. A lot of people do not realize that 80% of our planet is actually the deep sea. And of course, the deep sea is one of the most mysterious and understudied environments. But even though we don't know much about the dynamics of what's happening over there, we know that life thrives there, and there are so many amazing critters there. And I just want to share with you some amazing photos of these deep sea animals, and being the most abundant ones, deep sea fishes or deep sea crustaceans and deep sea cephalopods. And one of my main questions in my research is to answer this: how does the genomic diversity of deep sea species change over time? And in this presentation, I'm gonna focus on a few generations ago, specifically because I'm investigating genomic diversity in the years following the Deepwater Horizon oil spill. For those of you who don't know, the Deepwater Horizon oil spill is one of the major ecological disasters in the US history. It happened in 2010 in the Gulf of Mexico and is represented by an oil platform explosion that happened at 1,500 meters of depth, and it lasted 87 days, releasing more than 100 million gallons of oil directly into deep sea communities of the Gulf of Mexico. And I'm very lucky to say that I'm part of a group of scientists that has been investigating these deep sea community in the years following the Deepwater Horizon oil spill. So there were many iterations of this project, and we've been out at sea for many research expeditions. And this represents the longest time series sampling of the pelagic deep sea in the scientific history. And one of the things that we already know is that abundance of many taxa crashed over the years. For instance, a few taxa, they declined almost 80% when we compared the years right after the oil spill to nowadays. And my main question in this presentation is, can we detect any changes in genomic diversity over the recent years? So to answer this question, I'm investigating some of the most abundant species in the Gulf of Mexico, represented here by 2 deep sea shrimp and 3 deep sea fish. I analyzed samples from 2011 until 2023, And as you can see for the fish species, I'm analyzing a time frame of 12 years, but for the crustaceans, we just started collecting data for genetic analysis starting in 2015. So for them, I just have a 8 years time period. And I'm using ddRED-seq to investigate genomic diversity. So ddRED-seq is a reduced representation genome sequencing method. And of course, you can imagine that we do not have reference genome available for many of these deep sea species. So I'm employing a de novo approach. After preparing my libraries and sequencing all of them, I, of course, start doing all my filtering strategies. And what I did was I alternated removing low quality loci and low quality individuals, mainly using stacks. I'm not going to go through all the details, but here are some of the parameters that I used. And having the final data set in hand, I calculated multiple genomic diversity metrics for each year. And of course, for sake of time, I'm not going to present all of them, and I'm going to focus on effective number of alleles and expected heterozygosity. Once I had these metrics, I investigated the association between genomic diversity over the years to understand how genomic diversity changed in this time frame. And what I did, I compared multiple polynomial regression models, so a null model, a linear model, a quadratic model, third order, and fourth order model. So let's jump right into the results. This is my final data set. You can see the number of individuals that I analyzed for each species, the number of SNPs that I had in my final data set, and the time period that I analyzed for each species. And this is a summary of my results. As you can see, the species are not showing the same trend over time. Some of them are showing a decline, some of them are showing a moderate decline, and some of them are showing no changes over time. So to discuss the results, I'm going to first talk about the fish and then about the shrimps. And before actually talking about the results, we have to talk about diel-vertical migration, or DVM. DVM is a common feature in deep sea species. It is the largest migration on Earth. It happens every day in every ocean, and many deep sea species participate in this big migration. To represent DVM, I'm showing you here 4 different plots from 4 different ocean basins. On the x-axis, you have time of the day. On the y-axis, you have depth. So DVM represents the movement of individuals from deep waters to shallow waters during the night, and from shallow waters to deep waters during the day. So like I said, this happens every day, and many deep sea species engage in this behavior. So let's go back to our fish species. One thing that we know about them is that they are all strong diel-vertical migrators. However, when we look at the first one, Diaphis domerili, not all individuals within the species are engaging in this behavior. And in fact, the adults, they shift their lifestyle to a benthopelagic lifestyle, so they are more associated to the ocean floor, and the juveniles are the ones migrating up and down. In the case of the other 2 species, both adults and juveniles are performing the diel-vertical migration. Going back to Diaphis and knowing about about their natural history, one thing that we can speculate is that, so remember, the adults are the individuals that are associated to the ocean floor, not migrating. And of course, the adults are the individuals that are reproducing and contributing to the genomic diversity of the following generation. So we can speculate that maybe adults were more exposed to the oil spill than the other 2 species. That's why we see this decline in the species, that it's magnitudes higher than for the other 2 species. But if we just think about DVM and we compare just the other 2 species, we see that they are kind of different. So we have to look at another biological trait. So I looked at reproduction. For the first 2 species, they reproduce once per year. But when we look at the third one, Lepidophanes, this species reproduces multiple times per year. So we might think that in the same 12 years following the Deepwater Horizon oil spill, Lepidophanes had more generations than the other 2 species, which might imply that if it was affected by the oil spill, it recovered faster, or maybe it was not even affected by the oil spill. But of course, we have to be very cautious when we are interpreting these results, specifically because we don't have data from before the oil spill. But in fact, no one has data from before the oil spill. Remember, the deep sea is one of the most understudied environments. So this is a caveat of our study. So, and moving on now to the shrimps. A major issue to understand what is happening with the shrimps is that we do not have as many information about their natural history as we have for fish. One thing that we know about them is that they are both diovertical migrator species. They are both spawning species, but their seasonal patterns are unknown. So once I got these results in hand, it's kind of hard to right away interpret them and understand specifically why both species are not showing the same pattern, or why even we see a different pattern that we did not see for the fish. But of course, this is still a work in progress, and we have some insights of what might be happening. So the first thing that I want to tell you is that overall, when we compare genomic diversity between shrimps and fish, genomic diversity for the shrimps are usually higher than for the fish, probably because this group has a larger effective population size than fish. And also the shrimps potentially have a shorter generation time. But one thing that we don't know in both cases, just so both for the shrimps and for the fish, is how connected the Gulf of Mexico is to sites outside of the Gulf of Mexico. We don't know, we don't have information about gene flow or population structure regarding this species. And in the case of Phorcosurgia grandis, the second species, we actually found a pattern that we did not see for any of the other species, that it's represented by these ups and downs. And in this case, we think that the sweepstakes reproductive success hypothesis might explain what is happening. But again, I'm still thinking about these results. It's still a work in progress. So this is what I wanted to share with you today. So I'm gonna end up my presentation with some take-home messages. So the first one is that I'm very excited to share these results with you, especially because this is the first investigation on genomic diversity temporal changes in deep sea species, in which we have multiple time points, in this case, from 2011 to 2023. And what we are finding is that the biological traits potentially play a very important role in driving temporal trends in these deep sea species. Like I showed you, probably diovertical migration and reproduction are playing a huge role, but potentially other biological traits might also be important. And future directions of this project, I'm still analyzing the data, focusing both on a few generations ago, but I'm also going to investigate genomic diversity changes also thousands of generations ago, so in more ancient time. So that's it. Thanks for listening, for tuning in, and thanks to all my collaborators and for the funding agencies. And I'm happy to take any questions that you might have. Thank you.
All right, so I will ask a question. So you had pointed
Great.
out that, you know, the major animal groups you've got down there are the deep sea fish, the deep sea arthropods, and the cephalopods. Can you tell us a little bit about why you chose the fish and the arthropods and not the cephalopods to look at?
So the cephalopods are abundant in comparison to other things, but when we compare them to fish and crustaceans, they are way less abundant. So our sample size was way lower. But also other people in our group, they had done some genomic diversity analysis with the cephalopods, but just from 2011 to 2015. So we have all of the specimens in our freezer, so at some moment we might get them and analyze everything.
So I have a question, really nice
Sure.
talk. So you don't have samples pre oil spill, but I'm wondering, can you get samples of the same species outside of the area that the oil contaminated and use that as like a control?
You mean other sites that were not affected by the oil spill? Yeah, that's what we are trying to do now. and get samples from a sample size big enough to make these comparisons. And ideally, it would be nice to have samples that were collected at the same time, so we could better understand the dynamics of what was happening, what is happening.
Yeah.
But even though we don't have the data, so there are people looking at abundance, we're looking at acoustics, biochemistry. So everything is indicating that the oil spill played a huge role, but also we always have to be very cautious to actually affirm that.
I'm sure it's very easy to collect deep sea samples.
I wish it was. Any more questions? All right, thanks everyone.
Yeah, let me just share my screen. I think it's visible. Yeah. Yeah. So good morning, everyone. It's too early morning here in India. So my name is Tithi Paul, and I'm an undergrad student of Vishwabharati, which is a central university in India. So today my topic is going to be evolutionary implications of zebrafish locomotion, insights from biomechanics and neuroecology. I basically worked on fin strokes movement of zebrafishes. As we all know that movement in zebrafish plays a very crucial role in pre-predation or foraging in case of zebrafishes. Therefore, fin strokes is a very crucial part of their movement. Now, the question is why arsenic? As we all know that arsenic is already established as a neurotoxicant, as it directly attacks to the brain behavior circuit of an organism. And I basically looked through it in zebrafishes in this project. And according to ATSDA Substance Priority List of 2022, arsenic stands as the #1 priority list. So it made me a little curious that why should not we use arsenic in this project? So according to Thakur et al., arsenic also has some crucial effects in health issues, or like it also has some carcinogenic effects as well. So next, moving to here is one of my friend. I call her Finny. As being a passionate painter, I thought why only people should have all the fun. So I made one portrait of my friend, and let's understand what kind of fins are there for fish movement. So there are total 5 types of fins. So starting with dorsal fin, it helps the fish in turning and like stops or sudden direction changes. Like it acts as the keel to keep the fish stable in the water, as we can see in the boats. Next, we have the caudal fin. It mainly helps in acting or like getting propulsive force during swimming. And next we have anal fin, which helps the fish to maintain their stability. And next we have pelvic fin. It also helps in stabilizing and slowing down when swimming through flowing water. And finally, we have pectoral fin. It helps in generating propulsive force and like lifting force, which helps the fish in swimming upwards or downwards. So I will directly move into my methodology. We can see some bubbles of arsenic over here, and definitely it was non-lethal concentration, and it was a chronic exposure. And as I already said, that arsenic is already established as a neurotoxicant. So what we did, we had a set of 10 fishes, and we made a video of a control video actually of each fish from the set. And we definitely maintained a 12:12 day-night circle, and like we acclimatized the fishes at first. And then we recorded the video for each fish for 60 seconds. And then we counted the fin movement or the beating of fin, each 5 types of fins in a row. And we noted down the number counts. And after that, what we did, we used 1/50 LC50 dose of arsenic trioxide. And we got it from Sharkar et al. 2014. And we dosed all the fishes for straight 7 days. And after 7 days, again, we repeated the same methodology and counted the fin strokes of each type of fins. And we got some really interesting results. So here are my results. We can see a significant increase in number of fin strokes in all types of fins. And this is an average result of 7 days data. And we are definitely working on it to use other doses as well and using other contaminants as well. So yeah, this is a very preliminary data, but the results were really interesting. So what we can suspect from this result? So I think this study clearly indicates that arsenic is acting as an abiotic stressor or causative abiotic stressor in this over on zebrafishes actually. And as arsenic is attacking the neuronal activities, which is also altering the muscular activities of the fishes, which finally causing in the alteration of their fin strokes, as the muscular activities is related to the fins. So I think this is a very a significant result we can see. And yeah, this project was a combined project of ecotoxicology, neurotoxicology, and biomechanics, and definitely animal behavior as well. So yeah, this was all about my preliminary results of this project. And yeah, we are working on it. So yeah, thank you so much. Thank you for having me here in between all of you. And I'm ready for any kind of questions you may have.
So Taithi,
Yeah.
I have a question.
Yeah.
It seems like the fin movement was faster across these fish, and your slide here suggests that they're acting like Michael Phelps. So were they actually swimming faster, or were they just kind of jerking around? Did the arsenic exposure lead them to actually swimming faster?
Yeah, it was making them swim faster actually, because comparing both the videos, for the of the control video and the treated ones, they were swimming very fast actually. Like I compared their fish fin movement of 60 seconds and then after dosing, and they were moving very fast and beating the fins very rapidly. Yeah.
Very nice. So what's your next step?
Yeah, so as I said that I have treated them with arsenic trioxide as of now, and it was 1/50 dose. So I think I will try 1/10 and 1/30 as well for arsenic. And then I think I will try for other contaminants maybe not like only contaminants. I can try other, I don't know, maybe calcium or some antibiotics as well. But yeah, arsenic caught my eye actually while I was thinking of this project. So yeah.
So you had mentioned that this dose of arsenic is getting them to move faster, even though arsenic is toxic to them. Do you have a sense of how much more arsenic they'd need to get for it to be lethal, or is this just unpleasant for them, but they can survive much more of it?
Okay, so I took this dose from a project of Sharkar et al. 2014, where the dose I took was non-lethal to the zebrafishes, and I took 1/50 of the LC50 dose. So I was hopeful that this will not be lethal to them. So yeah, I took the dose from there, and it was 17.5 ppm.
Interesting. So I'm wondering if fish that are exposed to arsenic lower doses have issues related to mating or other sort of behavioral traits. Have you thought about doing behavioral assays?
Yeah, actually, what my aim was like to to establish the fin strokes as a cost-effective biomarker of environmental pollution monitoring or something. And as I have done this all manually, and it was so hectic to count all the fins, and like it took me around 10 hours for a single dose. So yeah, I think to maybe generate some algorithm to make this automated, or like to make it faster, and I can then do like put doses of different doses and different contaminants. So yeah, I thought about the fin strokes as of now, but yeah, I think that there can be significant changes in other behavioral aspects as well.
So you had noted that this increased both the speed of their swimming and the speed at which they're moving their fins. How large of an area do they have to swim around in, or are they just going in a fairly small circle in their container?
Okay, so it was a rectangular container, and like there was 9 liters of water, but like I mean, I just forgot the question. Can you just repeat?
Basically, how much room do they have to swim around in? Like, are they just swimming very fast in a circle? Are they making more complicated patterns when they're trying to get away from this concentration? What's going on with their movement?
Okay, so visually, I could see that they are moving faster, but when I was counting the fin strokes, as well, I was really concentrating to the fin strokes. I took each of the fish to another container to give them space and like record the videos of each fishes. So I think they were getting enough space and they were not haphazardly moving so fast. Yeah, and this faster movement was while they were in the tank with other 9 fishes, and where they were I was recording video individually of each fishes. So yeah, I could see faster movement.
Heidi, I have one more question, if you don't mind. So this is really interesting. So you're exposing the fish to the arsenic. Have you thought about, getting fish that have lived for many generations exposed to arsenic and see if there are any behavioral changes or fin movement changes that evolve?
Not really, I haven't, but I think this can be a really interesting issue. And what my main focus is, as I said, that like arsenic contamination is a very concerning issue in country like India, and like using this zebra fishes can be cost effective, as I said earlier. And like if we can see specific increased fin movement, like we need to establish all those algorithms and like patterns that how increment we can see in a fish and in a specific water region or water body. So yeah, I didn't see of a different generation who have lived in arsenic contamination. Like it was the first generation who experienced arsenic contamination for my research work. Yeah.
Very nice.
Yeah. Any questions? I think I finished so early.
Yori, if you want to start sharing your screen, we can get you set up.
What? I didn't get it.
The next speaker.
Oh, okay. Yeah.
Is my screen being sharing? Thank you. Okay, now I starting my presentation. Hello, I am Sasaki from Kyoto University in Japan. Today, I will be presenting on the theme Evolution of leaf vein patterns associated with habitat climate in angiosperm trees. Broadleaf trees, which are widely distributed across the globe, form the foundation of terrestrial ecosystems. One of the factors contributing to the success of broadleaf trees over conifers and ferns is the efficiency of water transport. Leaf veins play a crucial role in water transport within the leaf. Previous studies have focused on quantitative leaf vein traits, such as the length of veins per unit leaf area and the density of vein endings not connected to other veins. These traits are known to correlate with the temperature and precipitation of their habitat. However, the relationship between quantitative traits and the environment can vary depending on the species or region studied, sometimes showing weak or even opposite correlations. To understand the impact of leaf veins on plant adaptation to the environment, it is necessary to examine other traits of leaf veins. In this study, we focused on the branching patterns of leaf veins. Broadleaf trees have a primary vein running through the center, from which secondary veins branch out. These secondary vein branching patterns can be broadly classified into three types. Tree-like type have secondary veins that do not branch and reach the margin of the leaf. Looped type have secondary veins that do not branch and connect to an upper secondary vein. Combined type have characteristics of both, with secondary veins branching such that one reaches the margin while the other connects to an upper secondary vein. Theoretical studies suggest that water transport efficiency is higher in branching patterns without loops than in those with loops. Thus, the leaf vein pattern type may influence the plant's adaptation to its environment. In this study, we examined whether the leaf vein pattern type is associated with the climate of the distribution area. We conducted two analyses on 2099 species of trees to create a dataset and explore the relationship between leaf vein pattern type and the environment. For the dataset, we classified the leaf vein pattern types using images of leaf vein specimens provided mainly by the National Museum of Nature and Science. Then, for each species, we obtained habitat information from the Global Biodiversity Information Facility. This map shows the distribution of the species included in the dataset. Each point represents an individual, and the color indicates the leaf vein pattern type. As you can see, the target species are distributed globally. The dataset consists of 2099 species from 141 families and 666 genera. Additionally, we reconstructed a phylogenetic tree using existing data, consisting only of the target species. This database and phylogenetic tree were used for all analysis. In the first analysis, we examined the correlation between the leaf vein pattern type and the climate of the current distribution area, considering phylogeny. For climate factors, we used annual and monthly maximum and minimum values of temperature and precipitation, which are known to significantly impact plant distribution. The results showed no significant differences in precipitation among leaf vein pattern types, but significant differences in temperature, particularly mean annual temperature and mean temperature of the coldest month. The model likelihood was highest for the mean temperature of the coldest month. Since similar results were obtained for mean annual temperature and mean temperature of the coldest month, we present the graph for mean temperature of the coldest month. As you can see, tree-like types are found in the coldest habitats, followed by combined types, with looped type found in the warmest habitat. These results suggest that temperature, rather than precipitation, significantly influences the evolution of leaf vein pattern types. The tree-like leaf vein pattern type may function adaptively in response to low temperatures and related factors. In the first analysis, we found a correlation between the current habitat temperature and the leaf vein pattern type. Therefore, in the second analysis, we examined the evolutionary relationship between temperature and the leaf vein pattern type. Due to the constraints of the analysis, the data needed to be binary, so we classified the leaf vein pattern types into two groups: those with loops, combined and looped, and those without loops, tree-like. For the habitat climate niche, we classified climate based on the Köppen climate classification, dividing them into temperate, where the minimum temperature is below 18 degrees, and tropical, where the minimum temperature is above 18 degrees. Using the software-based traits, we performed ancestral state reconstructions with MCMC. We used two models: the dependent model, where the state of one trait affects the evolution of the other, and the independent model, where the two traits evolve independently of each other. We then performed model selection to verify the relationship. The result revealed that the evolution of leaf vein pattern types corresponds with changes in the climate zone of their habitat. A Bayes factor of over 10 strongly supports the independent model, and in this case, it was 28. We used diagrams to explain the evolutionary relationship between the two binary leaf vein pattern types and climate niches. These diagrams show the full combination of leaf vein pattern types and climate niches. The estimated ancestral states allowed us to calculate the frequencies of transitions from one state to another. The arrows indicate the direction and frequency of evolution, with higher numbers representing higher frequencies of transition. First, focusing on the looped type, we can see that its climate niche evolves with equal frequency toward both tropical and temperate zones. In both climate zones, the transition frequency from tree-like to looped type is high. This suggests that the looped type is adaptive regardless of the climate zone. Loops are known to be effective in maintaining transport functions. For example, even if veins are survived by insect feeding, water can still reach the leaf tip via alternative pathways in the looped sections. Next, looking at the tree-like type, we see that its climate niche frequently evolve from tropical to temperate zones. Compared to the looped type, which evolve with equal frequency in both zones, the tree-like type's climate niche evolve more frequently towards temperate zones, suggesting it may be adaptive in temperate regions. These results are consistent with the previous correlation analysis, suggesting that the tree-like leaf vein pattern type may function adaptively in response to low temperatures and related factors. In summary, the purpose of this study was to elucidate the evolutionary dynamics of leaf vein pattern types and identify the environmental factors driving the evolution. Our analysis indicates that the evolution from looped to combined tree-like leaf vein pattern types allowed for expansion into colder temperate zones. Some tree-like type in temperate zone evolved into looped type without changing their distribution, or moved to tropical zones and evolved into looped types. The looped type, which moves with equal frequency to both climate zones, appears to be adaptive regardless of temperature. These results suggest that the leaf vein pattern types have evolved along with changes in plant distribution areas. Thank you for listening.
Any questions for Hayori? Looks like one in the chat, which I'll read aloud. Katie Wagner says, really nice talk. Are there implications for the patterns you found in interpreting paleoclimate from fossil leaves?
Sorry.
Did you hear that? Do you want me to repeat the question?
Please, can you repeat the question?
Yes. Are there implications for the patterns you found in interpreting paleoclimate from fossil leaves?
Thank you for your question. I think it's very important to watch fossil leaves, but now I can't do.
Okay. One more question. Could you include the extent of herbivory in your model?
Moderate extent?
Could you include herbivory in your model?
Sorry, battery.
Herbivory. So plant herbivory by insects?
If you open the meeting chat, you can also read the question. That might be easier if you're having a hard time hearing it.
Thank you. Now I check the comment. Oh. However, your mother. This is very difficult, but because the herbivory's selection pressures may be different beyond the previous studies, so the value to use in models I can't choose maybe. Thank you for your question.
It was a really nice talk, Hayori.
Thank you.
Well, if there are no more questions, our last speaker actually withdrew, so we are done 15 minutes early. So this is the end of our first day with all the parallel sessions. You'll join us tomorrow morning. We start relatively early in the morning here on the East Coast, very early for those of you on the West Coast, kind of the afternoon for the people in Europe, with another Lifetime Achievement Award, this time for Ruth Shaw from the Society for the Study of Evolution. And we've got a whole day of parallel sessions after that. Thank you.